longtemps, on a postulé que les cellules naissaient par cristallisation. En 1850, Remak observe des divisions et affirme que toute cellule provient d'une autre cellule. Mais que sont les bâtonnets observés à l'intérieur du noyau? Fleming, en 1879, montre que ces bâtonnets, qu'il nomme chromosomes, se fissurent et se répartissent dans les cellules filles. Il donne le nom de mitose à ce processus. Mitos, en grec, c'est le filament. On y distingue l'interphase. Chromosomes et centrosomes se dupliquent. La prophase. La membrane nucléaire se rompt. Les chromosomes se condensent. La métaphase. Les chromosomes s'alignent au milieu d'un fuseau. L'anaphase. Les deux lots de chromosomes se séparent. La télophase. Les membranes nucléaires se reforment. Prophase, métaphase, anaphase, télophase. En fait, on est loin d'avoir compris cette danse.
With the beads, you didn't do it just with the chromosomes because it's not obvious. One of the things we are studying in the lab is how the chromosomes are moving during cell division, during mitosis, after you've disrupted the nuclear envelope. And this is a question that has really been studied for a long time. And this is an illustration of micrographs which are taken from 1882 by a German cell biologist at that time, Walter Fleming. And what they observed is that they thought that these fibers in the nucleus of the cell, they didn't know about chromosomes and the genetics of this. They observed that the arrangement of the chromosomes before and after cell division seemed similar. And so we observed the same process again, but now we don't have to kill the cells. We can film them when they are living and when they're actually doing the process of division. And that's really a big advantage.
Hi Daniel, I just recorded mitosis this morning, and I was interested in looking at the chromosomes and the dynamics of the chromatin. So I would like to know what I can do with this data to better study it.
Yeah, so we have a program that's very suitable to get a three-dimensional display of the raw data that you can see here. So first load our raw data set like that. So we will take another program that is designed to reconstruct and process. This is just one prophase stack.
Okay, here I can see the chromosomes already.
It's still very noisy, so we first adjust the contrast a little bit. So we look again, this was the original data stack. We can browse through it. We had only a very limited amount of slices, and when we look now at this one here, you can see it's much more, many more slices. It's a much higher resolution, and I hope that we can achieve now a much better reconstruction. So we have to rerun this one.
Okay.
And indeed, it looks much better.
So now we have an actual representation of the entire volume of the cell as it is going through mitosis.
Mm-hmm.
So now we can apply this to the entire mitosis and then follow what happens with the chromosomes.
We can project the data set, and we can now look.
So this is the cell
time-lapse
progressing through mitosis.
Exactly.
Then.
So now it's going to metaphase. We see the metaphase plate from the top. We can also twist it and look from the side. See how it congresses, forms the metaphase plate again.
And so we chose a technique now to look at where are the chromosomes during the process of mitosis, where we can paint the chromosomes different colors. This is achieved by lasers to paint one side of the nucleus in green and one side of the nucleus in red. And then you can follow how they change when the cell is dividing. And so when you do this kind of experiment, you can see that before mitosis, when you paint one half of the nucleus in green and the other in red, and you allow the cell to go through mitosis, after mitosis, it still looks very similar. The chromosomes are more or less in the same places in the nucleus than they were before the division. And this was quite surprising, because the more modern line of research has always assumed that during this movement in mitosis, that the positions of the chromosomes would be mixed up, everything would be mixed up, and you wouldn't have any order anymore. And so we observed that there is actually a lot of order where chromosomes go during the process of mitosis. Here you can see now an animation of this whole process where we start here. You can see how these chromosomes are moving around, how they are compressed by the microtubules, and the green and red mixes. And then as the chromosomes are segregating, the green and the red unmixes again. So most of the green goes to one side of the nucleus and most of the red stays behind on the other side of the nucleus. And we are trying to find out why they move non-randomly and why they don't just mix up during mitosis when all the membrane around them that usually keeps them in place is disassembled and they don't really have a reason to stay at a specific place anymore.
Nein, es war fast fertig, als du es aufgemacht hast.
Es ist abgestimmt.
Ja, ja.
Parce qu'en fait, pour les bactéries, tu peux juste les liser.
Ouais, je sais bien, mais je peux faire tout le travail. Je peux essayer ou quoi?
C'est ce que...
Of the nucleus. But I agree. I mean, the biochemistry and also the localization data would argue for the chromatin templating.
I have a basic question. When do most spores are assembled? Is it an interphase or right after mitosis?
You can also imagine that old pores just split into two, so that you have a doubling of the number of the nuclear pore complexes before the next cell cycle or the next mitosis.
But how can you imagine that? I mean, how can a spore just split into two? It's not like a cell just dividing. How can you imagine that mechanistically?
The next step we want to do is really look, as we did in the past, is look at very fundamental questions in cells. And I think the example we discussed on how chromosomes are positioned during mitosis is a good example because we examined a question that's more than 100 years old. And people really hadn't made much progress until recently when it became clear that the order probably was important in the nucleus. And so now we could study this in living cells and we could get new answers for how much order there actually is. And we want to go further with questions like this. So we would like to examine now in cells that are undergoing a change of fate when they have to differentiate what happens to the organization of the chromosomes inside the nucleus, to really ask, is this a functionally important order that we have observed, or is this just a consequence of the mechanics of the cell or how these structures are moved around?